Hello, Kobe here for Science Sapien, and today we're talking about the central dogma of molecular biology. This is in some sense a continuation of the molecular genetics presentation I put up, but this talks about how we go from DNA to proteins, which is a really important process of the cell, and that's called the central dogma of molecular biology. All right, so we're going to talk about how we go from DNA to proteins, and there's two main steps. There's one called transcription and one called translation. Transcription is going from DNA, deoxyribonucleic acid, to RNA, ribonucleic acid, which is another nucleic acid which serves as an intermediary, a message between the DNA and the proteins. And then translation is RNA to protein. So that's two different macromolecules, okay? So transcription is DNA to RNA. And both of these steps I'm going to be talking about three main things: initiation, elongation, and termination. So you're going to hear that over and over again. Initiation, elongation, termination. So really get that idea down because both of these steps have that. Okay? So the purpose of DNA transcription is basically DNA is too big to leave the nucleus. It doesn't fit through. So we want to transcribe DNA into mRNA, messenger RNA, which can leave the nucleus and go to the ribosomes, that's what's shown here, so that we can have a code to make proteins. So basically copying DNA to RNA, because DNA is too big to leave. What we're showing here is we're showing that process, which happens in the nucleus. All right. So three steps, like I said: initiation, elongation, termination. So initiation is the transcription factors. So there's a ton of proteins that basically tell the RNA polymerase where to bind. Because remember, we're only copying very specific genes. A gene basically codes for a protein. So let's talk about, for example, and we'll use this throughout the presentation, the gene that codes for the protein hemoglobin. Hemoglobin is used in the blood. Hemoglobin is pretty complicated, but let's just use that example now. So hemoglobin, let's say it has a gene, and that gene basically tells the cell how to make the protein hemoglobin, which carries oxygen in the blood. And this is an oversimplified example, so it's not particularly accurate, but it gets the message across. So there's transcription factors, these proteins in the nucleus, that bind to the beginning of the DNA sequence for hemoglobin. And they bind there, and then RNA polymerase, which very much like DNA polymerase, copies DNA into RNA. And it starts at a box called the TATA box. And the TATA box is basically a repeating set of TA, so thymine, adenine, TA, that initiates the start. So that's where the transcriptions bind on. RNA polymerase, binds on there and starts transcription. So that's initiation. That's how it starts. Elongation, exactly as it sounds. RNA polymerase unwinds and copies the DNA, copies it into RNA. And then finally we have termination. So the RNA polymerase reaches the end of the hemoglobin gene, or whatever gene is copying, and there's a very specific sequence of DNA that tells the polymerase, okay, stop. So the polymerase stops, and we get that entire piece of mRNA. So this is what we're seeing here, but it gets a little more complicated than just copying DNA into RNA. So we see here this blue. This blue is the initial DNA. There's a lot more on this picture than we're going to talk about, but basically here's the beginning. There's a TATA box. This is where the transcription factors, the proteins, bind on. So RNA polymerase knows where to start. So it starts copying here. It starts reading, right? Only one side. It only needs to copy one side, unlike replication. Reads, gets the termination sequence, and stops. So now we've gone from this DNA to this RNA. It doesn't have the promoter region. It doesn't have the downstream termination region. It just has something called exons and introns. And exons and introns are these segments of DNA. And basically, if we're talking about the hemoglobin gene is broken up. It's let's say it's broken up into chapters like a book, but we only want, let's say, chapters 1, 5, and 7. The rest don't apply to this hemoglobin. They apply to something else. So the exons are what we want to keep, and we want to get rid of the introns. And the way I remember exons keep, introns stay. Introns stay in the nucleus, and exons leave. Exons are used in the ribosomes to make proteins. So we have this primary RNA. We cut out the introns, which is called RNA processing. We fix any mistakes. And then we get this region right here that's just exons, just the stuff we want, just the chapters we want to make the protein we want, for example, hemoglobin. And then we do a little more processing. We add a 5' cap, which is just a bunch of phosphates and a guanine. And then we have a poly A tail. It's just a really long strand of RNA that's just made up of A's, adding these over and over and over and over and over and over again. And the reason we do that is to protect it. So right here is a cap, here's a cap, kind of like the ends of a shoelace. It protects the middle of the shoelace. It protects the mRNA. And then this mRNA, the post-processing mRNA, can leave the nucleus and go to where it needs to go. So again, just talking about processing. So it occurs after transcription, but before the mRNA leaves the nucleus. So spliceosomes are proteins that cut out introns, leaving exons for translation. Remember, those introns and extrons are different parts of DNA, segments that we either want to keep or don't want to keep. And then, like I said, we have a guanine cap, modified or GTP cap, so it's guanine and phosphates. That's on the 5' end. If you don't know what 5' end, go check out my molecular genetics video. It talks all about the structure of DNA and replication. And then we have the 3' poly A tail, so it's that long strand of adenine that's used for protection, recognition, transportation. So it's an ID tag. It's protection. It helps the RNA move around the cell. Very important. So we have to process this mRNA. Keep in mind, too, that an intron for one gene might be an exon for another. That's called alternate splicing. So if you imagine for a second, this is my segment of DNA. I'm going to break it up into 4 parts. Okay? Let's label them 1, 2, 3, and 4. Let's say one gene, gene A, or protein A, is parts 1, 2, and 3. So all I need to do is cut out part 4. But maybe gene B, which is slightly different, has parts 2 and 3 only. And protein C is 1, 3, and 4. 4. Let's finish the 4. Okay, so what this is called alternate splicing. We have the same 4 segments of DNA, but we're only going to need some parts for different proteins. So this is called alternate splicing because we have the same segment of original DNA, but the RNA is going to be made up of different parts, different introns and exons. So for example, A has exons parts 1, 2, and 3, and intron part 4, whereas C has exons 1, 3, and 4, and intron 2. So that's called alternate splicing. We use different parts of the same DNA to make different proteins. And this means DNA gets really complicated. This process gets really complicated really, really quickly. And you don't necessarily need a lot of DNA to have a lot of proteins. All right, so like I said, translation is going from DNA to RNA, and transcription is going from DNA to RNA, and translation is going to RNA to proteins. And the way I remember this, because they're such similar words, translation, we're changing languages. We're going from nucleotides to amino acids to proteins. So that's how I remember translation. We're changing language. Whereas transcription, we're in the same language. We're just using a different, font, basically. We're going from DNA to RNA. Same language, different font. Whereas translation, RNA to proteins, is a different language. We're going from nucleotides, nucleic acids, to proteins to amino acids. All right. Well, what are the 3 parts? Like I said earlier, we have initiation, elongation, termination. Same 3 parts, different steps this time, though. Okay, and the purpose of translation is we have this mRNA, but we want to make proteins, right? Proteins do all the work in a cell. So what we do is we go to the ribosomes. If you don't know what ribosomes are, check out my video on cell structure. Ribosomes are these little organelles that basically turn RNA into proteins, and ribosomes are made out of proteins and tRNA, or not tRNA. They're made out of rRNA, ribosomal RNA. And what they do is basically they take these tRNAs, these translation RNAs that float around the cell and connect to amino acids. There's 20 amino acids known to nature on Earth. And there's tRNAs that match up to each of these amino acids, and the tRNAs can communicate with the mRNA and grab the right protein. All right, so we have something called a codon. So if we think about a strand of DNA or RNA for a second, and each one of these is a base, what we can do is we can break each of these bases into groups of 3, like 3-lettered words, and each one of these is called a codon. Codon 1, codon 2, codon 3. Okay? And there's these units of 3 bases, and it's kind of like a word. They each code for a single amino acid. This is where the translation comes in, right? We're going from groups of codons, groups of 3 nucleic acids, and each one of these codes for a single amino acid. So, for example, the code AUG, adenine uracil, which replaces thymine in RNA, and guanine codes for a start codon, a methionine amino acid, and this starts all proteins, okay? That AUG codes for a very specific amino acid. Oops. So we have anticodons, which are on the tRNAs, the transfer RNAs, and they're the opposite 3. So if I had AUG, we would go UAC for the anticodon. And the tRNAs basically have these anticodons on one end, and on the other end they connect to the amino acid. So for AUG, they connect to methionine, the start amino acid. Okay? tRNAs are kind of like... basically what they do is they carry around amino acids and have a code that says, hey, look, I'm carrying this amino acid. That binds to the mRNA, and we get the right sequence of amino acids because we have the right sequence of codons and anticodons. All right, so now we're going to look at the ribosome. The ribosome has these 3 parts. The A site, the P site, and the E site. A is entering, E is exit, P is where we add stuff on. So there's 2 subunits. There's the large subunit and the small subunit. And basically when the mRNA comes in, they stick together. And the mRNA is stuck between them. And the 2 subunits are made of proteins and rRNA, like I said earlier. And the A site, right, enters. It holds the initial tRNA. P site is where the tRNAs grow. And then the E site's where they exit. Right, so there's 3 steps. First one, initiation. So the mRNA and the tRNA bind in which site? The A site. And we basically start pulling the mRNA through. Like a little strand of paper, we're pulling it through, and the ribosome's reading it. And it starts when it reads AUG. That's like the front cover. It's the book. It says start here. So that's the start codon. So when the tRNA says that, it binds on. It has methionine in the A site, and then we're going to pull it in the elongation. We're going to pull whatever tRNA is in the A site to the P site. Then the next codon, the next group of 3 nucleotides on the mRNA are going to be exposed in the A site. So the next tRNA that matches up perfectly with the anticodon comes in, binds, and we have another amino acid on the top end of that tRNA. And the 2 amino acids bind, and then we pull it another step. So the first tRNA is now in the E site. It falls off, but it leaves its amino acid connected to the second tRNA, which is in the P site. The tRNA in the P site is still connected to the mRNA, and it has 2 amino acids now. And now we have a third codon, a group of 3 nucleotides, exposed in the A site. The next tRNA is going to come in, attach its amino acid, and we're going to keep going and going and going until we terminate. There's a very specific stop codon. When that gets the A site, there's not going to be, the tRNA that binds is going to bind and say, okay, I'm done, I don't have an amino acid. The amino acid chain that we've created in the P site is going to fall off, and it's going to fold into a protein. Okay, so let's look at this again. The picture. So, right. We've got the ribosomes floating around in the cytosol, the fluid of the cell. Okay? The first tRNA comes in from the cytosol, and it matches up with these, what's this called? This is called a codon, right? And the tRNA has its very specific anticodon right here. This is the tRNA. So this is just RNA folded funny. And then on the top it has the amino acid that connects or that matches up with that codon and that anticodon. So let's look at this again. So first, we have a tRNA that comes in, right? This is my really bad drawing of a tRNA. Comes in, binds to the A site. When it's bound there, it's going to look like this. It's going to have an amino acid, it's going to have an anticodon, it's going to match. These two amino acids, this one and this one, are now going to bind. We're going to move everything over one. So this tRNA is going to go to the E site and float away. So we've lost a tRNA now. It's now floating back in the cytosol. It doesn't have, but what doesn't it have? It does not have an amino acid anymore. It's left it in this chain. Whatever's in the A site is going to go to the P site. We're going to connect those two amino acids, and a new tRNA is going to come in with its own anticodon, its own unique amino acid, come in, bond, and we're going to eventually grow this amino acid chain further and further and further until we get to a stop codon. The tRNA comes in, says, I'm done. The polypeptide, the chain of amino acids falls off, and we get a protein folding. So that's the central dogma of molecular biology. Transcription and translation, turning DNA into mRNA, to RNA, mRNA, and then to a protein. Thanks, guys, for watching today. If you have questions, leave them in the comments below, and don't forget to rate and subscribe. And as always, stay curious. And as always, keep questioning.